Hey everybody, I hope that you're having a good evening. What we're going to talk about tonight is bioinformatics, or what we're going to talk about in this video is bioinformatics. Now, when you hear the word bioinformatics, you might think,
Nerd alert!
or you might think, or you might think,
now that's where our geneticists take over. Thinking machine supercomputers and gene sequencers break down the strand in minutes. And virtual reality displays show our geneticists the gaps in the DNA sequence. We use the complete DNA of a frog to fill in the holes and complete the code. Whew. And now we can make a baby dinosaur.
but rather than think any one of those things, what I would like you to think whenever you think bioinformatics, I'd like you to think, what an extremely powerful field, what an extremely powerful skill set and tool set. What we're going to be focusing on in this is we're going to be focusing on bioinformatics tools that are centric or focused on analyzing protein sequences. Now, bioinformatics is a fairly wide open field in terms of what you focus on, but we're going to be focusing on protein sequence rather than focusing on or discussing nucleic acid, like RNA analysis or anything like that, and whole genome analysis. We're not going to get into that. We're going to be investigating 3 different tools that will focus on protein sequences. All right, so this webpage that I have pulled up right here, blast.ncbi.nlm.nih.gov, is the URL, or is the site that you can visit if you want to do something known as a basic local alignment search tool, or use the basic local alignment search tool. This is commonly known as a BLAST search, and basically what you can use this for is to put in a sequence. Now there's a couple of different BLAST tools. There is BLAST-N, which is for nucleotides, and BLAST-P, which is for protein sequences. And then there are different variations of it. So BLAST-X is a search for or search of protein databases using a translated nucleotide sequence. What we're going to do is we're going to use BLAST-P, which is a program which searches protein databases using a protein sequence. So what I'm doing is I'm inputting a protein sequence to align against a protein sequence. So there's a couple of things that you want to do with this. I've gone ahead and I've copied and pasted the sequence that I got from the worksheet that y'all are going to be doing. I don't really need to change anything with respect to my query subrange or anything like that. Likewise, I don't need to title my job because this isn't something that I'm going to save or need to pull up anytime in the distant future. My database, what I'm going to do is I'm going to leave this as non-redundant protein sequences. I'm not going to expand it. Like, one thing that you could do is you could look on the protein databank, in which case the protein databank is the database that you would be using. The protein databank is a repository of protein structures that have been resolved. So if you submitted your search to the protein databank, then you would only be looking at the catalog of protein structures that have actually been solved. So that's one kind of, I don't know if I want to say limitation necessarily, but that you'd be narrowing your scope. I'm not going to do anything with respect to changing my algorithm or anything like that because I don't really want to, and in part I don't know too much about what these different searches are. A BLAST-P search does not take a lot of time, so I'm just going to click BLAST. And just like that, it will take a little bit of time, but I will ultimately get a result from this. So here we are with our result. Now, I never titled my job or anything like that. It's simply protein sequence. I was given the job that I submitted, the search that I submitted, ultimately got a identifier for it, and that would be this A20SR6JK014. I don't really need to worry about that. This search is going to be preserved for about 2 to 3 days, or close to 3 days. But the rest of this chunk at the top is really just telling me something about the sequence that I made, or the search that I made. My search sequence was 238 amino acids long. Now what really is kind of important, or what is arguably the most important part about this, is what do your results show? Now, in the bottom half of this page, what I see is a very long list of, well, a bunch of things. And one of the things that I see is I've got all of these different options that are selected. And the first thing says green fluorescent protein synthetic construct. It has a maximum score of 496, a total score of 496, query cover of 100%, an E value of 3 times 10 to the negative 177. It's a lot of decimal places. And then percent identity. Now, currently, all of these are all of the sequences that came up that match up with my protein sequence that I submitted. Now, what you're most concerned with whenever you're looking at any sort of sequence result is your E value, or sorry, what I would argue that you're most concerned with is your E value and your percent identity. Now, what your percent identity tells you is basically of the 230 amino acids that you submitted as a part of your search string, what percentage of those are identical from your search string to this result. So this top result, I've got 100% identity. So every single amino acid in this result is identical in placement and everything to my search string. My E value, that number 3 times 10 to the negative 177, one thing that I want you to recognize about this is as you scroll down the page, one thing that you'll notice is your percent identity, and we're not sorting by percent identity, but your percent identity is going to go down from 100%, but that E value is going to go and get larger and larger and larger. So effectively, what I want you to take away from that is the larger that your E value is, or sorry, the smaller that your E value is, the better. Now, with that said, some of these, like these first 4 sequences, we've got a percent identity of 199.16, and E values that go from 3 times 10 to the negative 177 all the way to 6 times 10 to the negative 177. Now, whenever I click this first result, what that's going to show me is it's going to show me the identical residues. So what I've got in my first result, green fluorescent protein synthetic construct, I have 238 of 238 residues are identical. Now, what I've got is my query string right here, the first 60 amino acids, and then the subject that I get out of this. My query versus my subject. And what I see is every single one of those amino acids lines up. Now, if I click this link right here for sequence ID, that does not take me to the protein sequence that I submitted, but rather that takes me to the protein sequence of this result. Okay, so I can click that, and this takes me to an NCBI entry, right, and specifically a GenBank entry for this particular protein. This protein is the green fluorescent protein, mitochondrial protein. It's a synthetic construct. It's 247 amino acids. Well, my protein that I submitted was 238 amino acids. But what is worth noting about this is that the 230 amino acids that I submitted were identical to 238 of these 247 amino acids. Okay, now I'm going to jump back over here, and what I noticed, if I go back one more time, well, I'm going to go, sorry, my description. One thing that I noticed was my 5th result, my E value was larger, but my percentage identity was 100%. So I'm going to click this one. And what does this tell me? This tells me that my 238 amino acids out of 238 amino acids were absolutely identical. There was no sequence variation there. Whenever I click this sequence ID, well, this takes me to the page for that, and it gives me this entry for, well, a green fluorescent protein in basically every single place that this green fluorescent protein has been cited. And so this is a very lengthy entry and maybe not exactly what we're looking for in terms of getting a particular sequence or analyzing a particular sequence. So when you're looking at a accession number, when you're looking at a result that has an accession number or a description, that says something like rec name, that maybe isn't a good resource to go after, because that's, as was the case with this one, we're looking at something that's a little bit more of an umbrella sort of category, because basically whenever we click this one, this rec name, that brings up a huge number of sources and a huge number of kind of resources related to this particular protein. So if we click a different accession number, for instance, if we click this top accession number right here, well, what we're going to find is that this is going to give us a little bit more information about maybe one of the proteins that our protein is very, very similar to. So we can learn a little bit more about one particular locus of this. For instance, the source of this green fluorescent protein, well, it was a expression of a green fluorescent protein from a recoded gene inserted into Saccharomyces cerevisiae mitochondrial DNA. So this is an entry that you can look and find a PubMed entry, which I'm going to click that PubMed entry, and that's going to take me to a paper that this specific sequence was, I guess, used as an entry for the first time. So this was expression of green fluorescent protein from a recoded gene inserted into Saccharomyces cerevisiae mitochondrial DNA. And here's the abstract for that paper. You could click that paper if you have free access to that. If you're behind a VPN or logging into the library's web page or something through that, you can access that paper. Okay, so after NCBI and BLAST-P, there are other really great resources out there. One great resource that I think is available is this one known as XBASI. This is a resource, or XBASI is a great hub because it connects you to a number of different outlets and a number of different tools. If you look on the left-hand side of the screen, you'll see proteomics, genomics, structural analysis systems, biology, evolutionary biology, and so on. A number of different outlets where you can find some information and basically access a whole host of different tools. As you look with genomics, well, here are all these databases as well as tools that fall under genomics. If you're looking for a sequence alignment, characterization, or annotation of a gene or a potential gene, you'd want to go use one of those other tools. If you're doing proteomics like we're doing, well, this gives you, again, a large number of tools and a large number of databases. I'm going to zoom in a little bit so that you can see your proteomics kind of under the proteomics umbrella. There's protein sequences and identification, proteomics experiments, functional analysis, sequence sites, protein modifications, protein structure, protein interactions. I think that the protein interactions is a very cool tool or very cool collection of tools and databases, because it potentially gives you a way of looking at investigating how proteins interact with one another. And if you're talking about, or if you're investigating how a protein kinase functions, well, you can see what are some of the interacting partners of that protein kinase. Does it interact with and phosphorylate another protein, or does it simply make contact with? So those are the kind of cool networks that you can do a little bit of. You can plug your sequence in and analyze it, or just try to look at some of those networks. Now, what I want to do though is I want to go to protein structure, and this brings up, or whenever you go to protein structure, well, your tools and your databases, we're going to be looking at tools and some of those particular tools. Now, the tools that I want to look at have to do with our protein's secondary and tertiary structure. And so one of those tools is the CPH models. protein homology modeling. So what this is going to do, and I've actually already done this one, so we can save a little bit of time here. This is the CPH Tools webpage. This is a protein modeling or protein homology modeling server. The template recognition is based on profile alignment guided by secondary structure and exposure predictions. So you plug your protein sequence in. Nope, that's not my protein sequence. I already searched for that. So here is, just a second, here's my protein sequence. Let me go back over here. Plug it in, and this has a series of this has tools that it uses in order to ultimately predict something about a protein structure. So I'm going to click submit, and just to save us some time, well, here's my job. It's being queued and everything like that, or it is queued. And what I see here, this is the result that you would get. What I see here is I've got all of these different entries and scorings that will ultimately give me this result. Okay, now this result, the next tool that I'm going to go after is, and the final tool is again on XBASI in the Bioinformatics resource portal. It's this one, CFSSP. This is a protein secondary structure prediction tool. I click that. I'm going to go to this, and if you look at the top, it will tell you what CFSSP stands for, and that is Chow and Fastman Secondary Structure Prediction Server. So whenever you first talk about secondary structure of proteins, you talk about the Chow-Fastman prediction model. What Chow and Fastman did was they did this analysis back in, I believe it was the early to mid 70s. No, it was the mid to late 1970s, and they analyzed proteins. They analyzed amino acids, and they scored amino acids based on their likelihood to be found in a secondary structure, so an alpha helix or a beta sheet. So they scored every single amino acid, and ultimately that gave way to a mechanism to make a prediction. Oh, lo and behold, here's the paper, the second reference at the bottom of the page. Chow and Fastman conformational parameters for amino acids in helical, beta sheet, and random coil regions calculated from proteins in 1974. Okay, so it was kind of, I don't know, I wasn't right. So what you can do with this is you can put your protein sequence into this tool and click predict. And when you click predict, it's going to hit you up and say, nope, you got to enter the sequence in FASTA format. Okay, FASTA format. This is something that is it's a very subtle thing, but it's necessary for the tools to understand what you're actually inputting. And the way that you do it is with this open alligator mouth and then type in protein sequence. Click predict. Oh, I forgot to put a protein sequence in there. So protein sequence, post or copy, and then click predict. And here you go. Your output, one thing that you get is this graphical model. And that graphical model shows you basically the length of the protein from the N-termini down here, amino acid 0, all the way over here to amino acid number 238. And there's a color coding for every single amino acid based on its likelihood to be found in an alpha helix, a sheet, a turn, or a coil. And you see that same sort of thing down here. So here's our query sequence. The next line below that is likelihood of being found in a helix. There's an H next to those amino acids. Below that is an E, whether or not an amino acid is likely to be found in a beta sheet, a T for a turn, and then ultimately that information's kind of summed up and simplified in this line right down here, which is structure 1. So basically it's telling you of all the data that's there, what's likely to be found. So if you look at this region right here, we've got helix, 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 but we've also got sheet, sheet, sheet. And as it turns out, the more likely outcome of those two is for a beta sheet to be found in that region. So what this gives you is it gives you some percentages at the end. So total residues, there's 154 of the amino acids of our 238 238 amino acids that are going to be found, or likely to be found, in an alpha helix, 142 that are likely to be found in a beta sheet, and 34 that are likely to be found in a turn. Okay, now then you can break that down into percentages, and the great thing about this ultimately is it gives you potential information. It's not going to say concrete, yes, this is absolutely what's here, but it gives you some insight. So then you can look at something like this region right here, right around 175. There's one amino acid that seems to be in an alpha helix. What's the likelihood that there's an alpha helix here? Pretty slim, because it's only one amino acid that has a propensity to be in an alpha helix. That is surrounded by a bunch of amino acids that are likely to be found in a beta sheet. So there's a slim chance that you actually have that alpha helix there. If there is a secondary structural motif there, chances are it's probably going to be that beta sheet. At any rate, these are all the tools that I want you to think about whenever you think about bioinformatics. This result might seem totally bonkers. What the heck does it mean? Well, what this all means is, as we go through, sorry, just a second, what this is going to give us is an analysis and a comparison of our sequence, our submitted sequence, to data that is in the protein data bank. And so what we have at the bottom here is kind of what we care about, or what I understand what we're going to be looking at here is our query, our search string that we entered, compared to our template. So what this tool did was it used our protein sequence and looked at the protein data bank and said, all right, where's the protein that most aligns with the submitted sequence? And so if you look, well, we've got this comparison where we've got a lot of identical residues, and the entry right here is ultimately something known as, or something identified as 5HW9. Now that is a 4-character code that is commonly used by the protein data bank. So if you do a PDB protein data bank, the RCSB protein data bank homepage, if you go there and then you type in that result that we got, 5HW9, punch that into the protein data bank, and lo and behold, it's going to give me this right here. 5HW9, filamentous assembly of green fluorescent protein supported by a C-terminal fusion of 18 residues viewed in space group P21. Okay, lots of that might seem a little bit daunting and overwhelming, but what's important about that is this is giving us ultimately a structure that we can look at from the sequence that we input. We already knew from our BLAST search that the sequence that we input was for the protein, the green fluorescent protein. Now we can spin this structure around, and this is, maybe a little bit more intimidating than we're looking for, but what we're being told by this tool right here is the protein that we put the sequence in for most matches the protein data, or most matches the green fluorescent protein in the protein data bank. So this gives you a way to look at a protein based on the primary sequence of a protein. This gives you a way to look at a protein's tertiary structure. So in your biochemistry class, one of the things that you talked about was the relationship between sequence and structure of a protein, and structure and function of the protein and everything like that. But this is based on data that's available, sequence data that's available. This is a way that you can determine, or not necessarily determine, but see if there is some similarity between a protein that you're characterizing and a sequence that's already out there. Now, our result gave us something that was 97.9% identical, but we're working with a green fluorescent protein. It's entirely possible you might have a protein that's only 30% identical or something like that. But you know what? That gives you some insight into that protein's tertiary structure. Now, the last thing that I want to say regarding bioinformatics is, aside from it being a growing field, it's a very powerful field because of the fact that what you're able to do is access databases with tons and tons of information. Like for the ever since the human genome was sequenced, or ever since the first genome was sequenced, there's just an insatiable desire to sequence everything, and get the genome sequences of everything, get the proteome sequences of everything. And there's kind of a limitation in the sense, there's a limitation into the value of more and more sequence data. Sure, it's valuable. It will always be valuable to gain more sequence information, gain more insight into a particular sequence. But at some point, you have to think you can kind of collect all that data, but if you don't know how to analyze it and you don't know how to do anything with it, well, then it's basically a waste. And so whenever what we were just looking at was tools that are readily available. We were looking at tools that have already been written. And, if you have any insight into, or if you know how to use any sort of computer programming language like Python, or I believe it's called R, or Ruby on Rails or something like that, then you're in a position where, especially since everyone in this class is a biological sciences related major, you have a scientific know-how and potentially a software development know-how where you can merge those two fields and two strengths that you have to develop a tool, tool that utilizes a freely publicly available database to analyze sequence information. Because that sequence data is always going to be produced, but you have to use it for something. You have to be generating the sequence information is almost like a rinse and repeat. New organism, sequence it again. New organism, sequence it again. Or sequence that new organism. Same protocol over and over again, but it's only when you actually analyze that data and have a program that will properly analyze that data that you're going to get a lot from it. So if you have any insight into those, then by all means get after it, because that's a great way to use your scientific know-how, and from a career standpoint, my understanding is you make a healthy sum of money. All right, well, thank you very much. I hope you have a good one.